Hello student, good afternoon. Today we will start the new topic or new cell organelle, that is structure of lysosomes. Structure of lysosomes. What is meant by lysosomes? The lyso means digestive. Soma means body. That is the digestive body. Are tiny membrane bounded vesicular structures of the cytoplasm. It performs intracellular digestion of the cell. Lysosomes was firstly reported by D. in 1955. Lysosomes occurs in most animal cells and few plant cells. The animal cells which are secretory in function, such as the pancreatic cells, leukocytes, liver cells, spleen cells, and kidney cells, contain large number of the lysosomes. The lysosomes are particularly numerous and large in cells. Such macrophages, which performs special digestive function. The lysosomes often remain distributed evenly in the cytoplasm. Structure of lysosomes. The lysosomes are generally spherical in shape. Its shape is spherical. But certain meristematic cells of plant roots contain irregular shaped lysosomes. The size of the lysosomes usually ranges from 0.2 to 0.8 micron, but may be exceptionally large in the cells of mammalian kidneys. The lysosomes are round vacuolar structure which remains filled with dense material and acid phosphatase enzymes. The lysosomes are bounded by a unit membrane of the lipoprotein. Lysosomes are bounded by a unit membrane of lipoprotein. The space of vacuoles of the lysosomes contains dense granular, vacuolated and homogeneous material. Different kinds of lysosomes of the kidney cells of rat, myelin fibers of the undigested feet, and granules vacuoles, kidney shaped lysosomes. Kinds of lysosomes. Polymorphism in lysosomes. In different types of cells and at different times in a same cell, 4 types of lysosomes have been observed. How many types? 4 types. 4 types of lysosomes have been observed. Namely, these are as follows. Primary lysosomes. Secondary lysosomes. Residual bodies. Autophagic vacuoles or cytolysosome. The primary lysosomes or pre-lysosomes are small sac-like bodies having many enzymes. These are either secreted directly by the granular endoplasmic reticulum or by the cisternae of the Golgi complex. The secondary lysosomes are also known as heterophagosomes or digestive vacuoles, When the cells feed on foreign or exogenous or extracellular substances by the process of phagocytosis or pinocytosis, heterophages, then the plasma membrane forms the membrane bound vesicles known as pinosomes or phagosomes around these substances. The pinosomes or phagosomes ultimately fuse with the pre-existing primary lysosomes and from the secondary lysosomes or heterophagosomes. The digestion of engulf substances takes place by the enzymatic activities of the hydrolytic enzymes of the secondary lysosomes. When the digested food of the secondary lysosomes, when the digested food of the secondary lysosomes diffuses into the cytoplasm, there remain indigestible material or debris. The secondary lysosomes are now known as residual bodies. In certain cells, the residual bodies remain in the cells for long time and play important role in the aging process. The residual bodies are formed due to lack of certain enzymes in the lysosomes. The residual bodies causes about 12 pathological diseases in man, that is fever, congested heart failure, hepatitis, pyelonephritis, hypertension, mitosis, joint injury, leukocyte, granules and tissue injury. The autophagic vacuoles are also known as autophagosomes or cytosomes. The autophagic vacuoles are formed when the cells feed on its intracellular organelles such as the mitochondria and endoplasmic reticulum by the process of autophagy. The autophagic vacuoles are formed in the spherical, spatial, pathological, and physiological condition. C.D. Dewey in 1967 and Allison in 1967 have observed that during the starvation of the organism, many autophagic vacuoles develop in the liver cells which feed on the cellular components. This is the structure of lysosomes. Okay, thank you.